So we're going to be covering neurofibromatosis 2. This is a genetic disorder caused by germline mutations in the NF2 gene. This is located on chromosome 22. So the way to read this is 22 is the chromosome, and then the Q12 is a location. So it's Q is one of the arms, one of the sections of one of the arms, and 12 is like a subsection in that Q area. So it's still kind of broad, but it kind of says this part of this chromosome is where the gene is located. And that can become important, especially when you look at the kinds of mutations, and even in NF2, sometimes entire chromosomes can get mixed up in cancer. And so what happens is, like, maybe they double append, or you cut off an entire section, you're just losing an entire part of a chromosome. There's all kinds of, it's not just point mutations of the amino of the base pairs. You can also have chromosomal damage structurally, and big level stuff that happens as well. So going back, the NF2, which is neurofibromatosis type 2, it leads to the development of multiple benign tumors, particularly vestibular schwannomas. We'll get into that, but vestibular is the ear canal, something to do with the hearing ear canal. Meningiomas, ependinomas, gliomas, and it encodes the Merlin protein, sometimes also referred to as schwannomin. The most common one that I see is Merlin. So unfortunately, this disease, it has too many terminologies, but you just learn them. There's only 3 of them. So NF2 is the gene. Well, I guess more than 2. NF2 is the gene name in short form. The protein it encodes is Merlin. The disorder, when you have this disorder, it is neurofibromatosis type 2, and then there is a specific type of that called Schwannomatosis, and that's what we're going to be looking at. So Schwannomatosis is a certain type of schwannoma, and we'll get into that, but it's a certain type of a tumor. Generally, it's going to be these vestibular schwannomas, which are tumors in the hearing canal, but once you have these tumors, they can be in other places in the body. So NF2, the NF2 gene is inactivated in most schwannomas, either by biallelic mutations, mutations on both of the alleles, or loss of heterozygosity on chromosome 22q. So that would be, a challenge where one of your chromosomes is damaged in that case. All right, so the NF2 schwannomatosis disorder, it's caused by a pathogenic variant in a gene on chromosome 22. So schwannomas are benign tumors that arise from Schwann cells in the peripheral nerves. Schwann cells are the cells that wrap around nerves and make the nerves propagate signals faster. So the Schwann cells they kind of have a fatty kind of fruit roll-up style thing that they do, where they wrap and wrap and wrap with this fatty tissue. And what that fatty tissue does is, when you have a signal going through a neuron, it is a propagation of sodium ions that are flooding into the neuron from the surface. There's these ion channels that open up, and they flood in. And what happens is, when you have the fatty insulator, it actually accelerates the propagation of that sodium wave. you actually have more of the concentration and more of the, because these are ions in solution, so there's actually a voltage that you have. You have a propagation of voltage through the nerves, and that the insulator just helps that voltage propagate faster. So what you have, why do you have Schwann cells? It's because it helps you transmit signals across your body faster. Very commonly, these are in the uh, peripheral nervous system, and for example, and also the central nervous system. But for example, your pain receptors, it's very important that pain gets to you very quickly. Those are going to be wrapped. Things that have to travel a long distance, so things in your spinal cord, they have these myelin as well, because you're just trying to get the information there very quick. So that's why you have Schwann cells. And so when we talk about a schwannoma, they're benign tumors of the Schwann cells, generally in the peripheral nerves. That's the emphasis here for schwannomatosis. And the tumors often originate from the vestibular nerve. So again, hearing is the vestibular canal. So there is a cranial nerve, which is one of the special nerves that goes from the top of your spinal column, I think. Actually, I can't remember if it goes directly to the brain or not. But anyway, it's a cranial nerve, and it's one of the very important nerves, and it is carrying audio information. So what you're hearing comes through the vestibular nerve. And so we're having these tumors in this vestibular area, and they can develop and they're commonly in the vestibular nerve. However, they can be anywhere from the glial Schwann junction up to the nerve terminations within the auditory and vestibular sensory organs. So I have a picture of this coming up, but it's generally on the vestibular nerve, but it doesn't literally need to be there. It can be anywhere in the surrounding area, any of that supportive tissue, just anything that part of the system is all going to be called a schwannoma. Although histologically benign, and again, histology is taking a slice of tissue and looking at it under a microscope with a stain. The most common way that you interpret cancers, you take a biopsy, and somebody's looking at that biopsy under a microscope and using their knowledge on the shape of cells relative to the parts of the body to know, does this tissue look right? Is it really messed up? Is there way too much stuff in it? These kinds of very technical things, but can also be a little bit of an art, studying these cell patterns. In any case, the histology tells you that it's benign. And although it's benign, vestibular schwannomas may cause hearing loss, tinnitus, the ringing of the ears, facial palsy, and when large enough, brainstem compression and even death. So the early symptoms typically are these hearing loss and tinnitus. You get this like ringing, and it's just not going away. You can't hear as well. That might be the first thing that you detect as a symptom. By that point, of course, they're already starting to grow and do things that you don't want. But they can get even larger, and then at that point, yes, can become fatal. And also, by the way, very painful. So they're often unilateral on one side of the head, but can be bilateral, especially in the NF2 syndrome. So we'll take a picture. So here is actually, let me start on the right. Here's the vestibular canal. Here is your cochlea. This is where the fluid in your ear is, and inside of that, there's these little hairs that detect the vibrations. And the deeper the hairs, I think I don't remember if it's the lower pitch or the higher pitch, but one of them, I think maybe say the lower pitch goes deeper in that kind of little spiral shape. And then back here are the nerves, right? And so these are the cranial nerves, the vestibular nerve right here. And then you have that schwannoma that they're developing somewhere in that area. You'll see that there is this, of course, the skull and the bone junction. This little tube right here is a fluid drain for draining fluid out of your ear. But somewhere in that area. So when we talk about unilateral or bilateral, we're saying one side of the head or both sides of the head. So when it's sometimes you have a mutation like the NF2 schwannomatosis, and you're just very susceptible to getting these tumors, and you're going to get them not just here, but in other places in the body too. You see these gray dots down here, other ones in other places of the body. But there's also a possibility that you just kind of spontaneously get a mutation, in which case it's going to be called a sporadic mutation. Sporadic means that you're just getting it after you were born. So maybe you were born without the mutation, and then at some point in your development you get a mutation in one part of your body, and that can make that area very susceptible to being a tumor. So if you have the NF2 gene mutations, you're born with that, but there is a difference between being born with the gene mutation and then sporadically getting that mutation a little bit later in life. So the treatment options, generally, there aren't very good outcomes here, as far as there isn't much you can do. A lot of it is just we're going to try a bunch of things. So you're going to try and protect your hearing, reduce neurological impairment. The most common is wait and watch. It's the observation. So we see that you have an issue. It's not thought to be fatal. It's not thought to be an issue. Maybe it's small. It's going to be worse to give you chemotherapies, which are toxins, and it's going to be worse to give you radiation. So we're just going to wait and see. Next up, we have the microsurgery. So when large tumors press on the brain regions, and there's maybe some more severe symptoms, then the surgery may be necessary. However, there's dangers associated with surgery, which include hearing loss, injury to the facial nerves, balance issues, and those can be particularly troublesome with the NF2 patients, where you have the need for surgery potentially on both sides of the body. There is radiation, so radiosurgery, gamma knives, that's an option as well. The schwannomas are, again, benign, made up of abnormal Schwann cells, which are cells that insulate the nerves. They can develop on any nerve in the body, including the cranial nerves, spinal nerves, and peripheral nerves. The most common tumors in NF2 schwannomatosis are the vestibular schwannomas. So probably a lot of what we're going to focus on are the vestibular schwannomas, but be aware that there are other types. And when you're reading the literature, you kind of want to get a sense for what type of NF2 are they talking about. So we're going to use this notation here, NF2-SWN. This is from the Children's Tumor Foundation, and it's a special focus on the NF2 schwannomatosis. And we'll get in, there's a chart I have coming up where we're like, what are these other ones? But that's the one that we're focusing on. It's mostly focused on that vestibular nerve. It carries sound and balance from the inner ear. It's the 8th cranial nerve. So as we talked about, you can also develop other types of tumors. So meningiomas are benign tumors of the membranes around the brain and spinal cord. These occur in 80% of people with the NF2 schwannomatosis disorder at some point in their lifetime, but they often don't always cause noticeable symptoms. The ependymomas, these are the benign tumors that develop from cells on the ventricles of the brain and the center of the spinal cord. So ventricles are fluid-filled sacs in the middle of the brain. If you ever heard of cerebrospinal fluid. You have a fluid flow, and so it cleans and nourishes. And there's the sacs in your brain, and there's a tube of it all the way down your spinal cord. And so that is obviously an epithelial lining. There are, it's a tube, and it does, fluid flow just like uh, your blood system, your arteries, or also your lymph nodes. They're all very similar connective tissues. And so you can get benign tumors in those cell linings as well, and that's about 20% of people. Um, and then gliomas are tumors that originate from glial cells. Those are the supportive cells in the brain and spinal cord. So most of the mass of your brain isn't neurons. Most of it is actually the glial cells that are supporting the neurons. Neurons are very static. They don't move much. You could have glial cells kind of moving around them, cleaning things up, providing nutrients. They've been found to play a role in some kinds of signaling, but they're generally just considered the maintenance and support cells. So it can also cause issues with vision loss. Most people develop symptoms in their late teens and early adult years, although 10% develop symptoms during childhood. So that's kind of an important point. Anytime you hear about like when a disease onset is related to mutations in cancer, when is the onset is very meaningful because it's telling you a lot about the developmental biology. So for example, some mutations are so severe that you're a non-viable embryo. Like we never hear about these people because they are never born, but or they're just not viable very early. The child onset is also meaningful. Maybe it's a very critical part of your system. The teen and adult onset is also meaningful. There's maybe implying a hormonal relationship that's not really studied well, or maybe it's something that takes some time to acquire. Like you're accumulating not just one mutation, but maybe you need a set of additional mutations to really trigger the cancer. There's a lot of reasons for this. So the most common first symptoms, as we talked about, was the tinnitus, the ringing of the ears, and hearing loss and balance problems. Other ones to be aware of, though, we have difficulty swallowing, seizures, vision loss, loss of balance, pain, muscle wasting. So this is known to be a very painful one to have. You have these tumors in the inside of your ears. It's very painful. You hear the ringing and you're losing your hearing, and it's not a fun one to have, in fact. All right, so we talked a little bit about the different types of schwannomatosis, and just be aware of this. This is going to play a really big role when you're doing the literature reviews, because you have to remember that an article is written probably with a hyper focus, and you have to know what focus they're focusing on. They may be, I've seen a lot of NF2 related to this LZTR pathway, and that's not the one that we're particularly focusing on, but it's important to be aware of it. So there's also this SMARCB1 pathway, and you can see here the incidence. The more common one for a rare disease is this NF2 schwannomatosis, 1 in 25,000 births, but there are people born with these even rarer alternative versions, the SMARCB related schwannomatosis, LZTR1 related schwannomatosis, and then there's these other ones that are just not well defined. Something to be aware of, and Olivia, this is kind of pointed at you, compared to NF1, this is an even more rare disease. So what that means is that there is even more challenge finding data. Data sets are smaller. The patient populations are smaller, and everything is small. The funding is smaller. Everything is harder, right? So these rare diseases, we're really taking on something very challenging to make progress. And even if you're the best scientist in the world, but it's hard to get good data sets, very expensive, we're going into this knowing that this is a very challenging thing to study on face value. With that said, any progress we can make is greatly appreciated because it's just really hard. It's very hard to make progress. We have that unique ability to push forward a field where a lot of these people that do have this disorder don't have a lot of outcomes that are good. It's very hard to give them help. Not a lot is known medically, and that's a challenge, right? So again, we talked about the NF2 gene carries the protein called merlin. Merlin is the mosin erzin redaxin-like protein. You don't have to remember that. I wanted to say it one time. It's here for reference, but that's what it is. Merlin helps prevent cells from multiplying too often. And without Merlin, cells would continue to multiply and become tumorous. So I think they would call that a tumor suppressor. Merlin is a tumor suppressor. It's like the brake in your car. And if you lose the brake in your car, then your car is likely to just go too fast and not be able to slow down. So you need that braking system. That's the role of Merlin. Without it, yeah, you can get this tumor development. So we did talk about a little bit about sporadic, and it's also sporadic again is when you develop the mutation after birth. That is in comparison to familial mutation. Familial means that it was inherited and you've had it since birth. You've had this mutation since birth. So about half of the people born with the NF2 schwannomatosis inherit it from an affected parent. And for the alternative one, the sporadic one, that would be the other half that did not inherit it from a parent, and they're often the first in their family to be diagnosed. When you look at those sporadic people, half of those people have genetic mosaicism. It's a situation where an individual is composed of two different genomes, two different cell types. And so you can imagine that, say you are a fetus and you're developing and you get a mutation very early in your development, maybe on your arm, you have some mutation. When you're small, it's just a few cells, but as you get bigger, that can become an entire tissue. And now you're a human who's made out of two related but different tissue types. And so that's the mosaicism. You have different mutations in different physical parts of your body. And so, for example, you could have that mosaicism in the area of the vestibular canal. Maybe you have some of that NF2 mutation on one part of your body, but not the other side, for example. And so that's half of people with sporadic have that. Go ahead, Anushka.
I don't have like a question or anything, but I remember we were studying about this in AP Bio and just like little fun fact if people did not know. Calico cats and like um tortoiseshell cats have their specific kind of color because of genetic mosaicism. Just why like you can see the different splotches of color because some of the cells, the X chromosomes are like inactivated or something like that.
That's true. Another really pretty example is sometimes you have really complicated flowers where like one of the petals is orange and the other is red. And that can be mosaicism too. You can have weird, it kind of looks like a mutant, but they can be very pretty. In the case of flowers, you have just kind of a variation in color phenotypes. Um, All right, so we talked about the treatments up to this point, and you notice what we didn't talk about was um, compound treatments, and that's because there aren't very good outcome. There isn't anything approved for NF2 treatments. So that's a problem, right? That's even worse than the NF1 situation where now they're up to 2 approved compounds. We're in a situation where there's 0 approved compounds. That's a really big challenge. So again, most of what is done is on that surgical wait and see side, or just working with the symptoms and trying to resolve the symptoms. So what I'm presenting here is not like the exhaustive representation of what is known clinically. In fact, I have not done a deep clinical trials investigation for NF2. That might be work that we want to do. But I want to give you guys a flavor for what is known. We talked about the surveillance, surgery, radiation, rehabilitation, implants, just genetic counseling. When we do talk about the therapies, though, we do have some clinical trials and off-label use. So I'm calling out here bevacizumab, which is an anti-VEGF, and it's thought to shrink some vestibular schwannomas and improve hearing in some patients. Tyrosine kinase inhibitors are something that we want to maybe take a look at. So brigatinib, for example, is something we might want to look back at. They're also talking about multi-kinase inhibitors. Maybe they go after more than one target, not just one thing. mTOR and PI3K pathway modulators, something to look out for. So you'll notice that these are kind of broad. There's only a few pointed examples. That's a challenge, but again, that's what we're here to do. Is there is a gap in the medical understanding and what some good targets might be. So we need to, again, this is not exhaustive. We need to do some literature research and kind of catch up with the field. However, there's no reason to think that there's any smoking guns out there yet. We would want to help push forward a story on what we think might be worth researching. All right, so as far as prior work, one thing I do want to call your attention to is that our colleagues at the Children's Tumor Foundation, who we're very close with, a few years ago, I think in 2017, 2018, did a large NF2 cross-lab symposium and collaboration, and it was called the Schwannomatosis Project. And that is like the best data set in NF2 right now. It's the most comprehensive, the largest, and it's something that we want to be aware of. So yes, that is something that we will be looking at. Just want to call that out there, in part because there's a history between our working group and the Children's Tumor Foundation. They previously funded us. We're also going to them again for potentially funding the student paper from last year. So we want to keep that good relationship going. They have a possibility to cover our publication costs this year too, if we go that far. So at this point, I'm going to move from the disease into more of the pathways, the cell pathways, the understanding, like what is it, what's it actually doing. Unfortunately, this is going to be kind of complicated, in part because it's probably not understood very well, but also because it's just complicated. So it's okay if you get lost here. That is why we are going to share the slide deck. And I'm happy to revisit this multiple times over our working experience, but I did want to do a fair first pass and just here's what it is, and hopefully it's helpful. So the Merlin protein, it's part of a set of other related proteins, the ERM proteins. I don't think that's too relevant. It works to regulate important pathways, the mTOR C1, the Hippo pathway, and the RAC-PAC pathway. Now, when I go into these pathways, and especially as you continue your cancer and biomedical careers, I don't think anyone expects you to remember every single pathway in the body. There's just too many, right? But there are some very commonly occurring ones in cancer. And in fact, these three are, like, if you were to talk to a professor or something, they would probably recognize these. Um, so the reason I'm showing you is so that you kind of start to make an association with it, like, oh, I think I've seen that somewhere, right? And that's all I'm expecting you to do. I'm not expecting you to memorize it, but I am expecting you to think, oh, I might have seen that somewhere. I think it had something to do with cancer pathways or something. Like, enough for you, like you can dig deeper if you need to. So again, I'm not expecting you to memorize these, but I do want to present them to you. We'll go through each one, and hopefully it'll make more and more connections for you over time. So the Merlin is regulating the mTOR, Hippo, and RAC-PAC pathways. So it moderates it somehow. It is also known to suppress signaling. So there's the ERB B2, ERB B3, and the EGFR. This is epithelial growth factor receptor. That's an easy one. It's a growth factor receptor. It's on the outside of epithelial cells. And, again, this is the kind of thing where if you had any damaged tissue or a cut, you have signaling molecules in that growth area telling you to recover. That's some of those are the growth factor compounds, and the receptor is this one. Epithelial growth factor receptor is often used as a representative for a class of receptors. There's a lot of different receptors. This is just a really well-studied one that represents a class of receptors. It has to do with what are called G-couple protein pathways, and they're just know that it's the receptor, and it has to do with signaling pathways downstream. So we're doing something to, again, suppress that signaling. So we're again modifying how these growth factor receptors are being handled, and how it has to do with cell regulation, cell survival, and cell division. Another thing about Merlin, though, is it actually can enter the nucleus. And there's specific cases when it enters the nucleus. This is actually a big distinction from when we were studying the NF1 disease. NF1 was not known to enter the nucleus. It was a very big compound. The Merlin, however, can, in some cases, enter the nucleus and inhibit some stuff. The E3 ubiquitin ligase, we'll get into it. But that's one of its known mechanisms. It's also playing a role inside the nucleus. So loss of the Merlin protein, or the degradation of it, leads to tumorigenesis. So that is the propensity for tumors to grow, right? So the day you lose it, you don't necessarily have tumors. Again, you lost the brake. You're not necessarily speeding yet, but you lost the brake. Very likely you're going to be out of control not too long after. A few of these growth factors hit your outside, and the gas is given to the cell. Well, there's nothing taking the gas off. You can get out of control very quickly. So we talked about it's on that chromosome 22. So the NF2 gene is inactivated in most, if not all, schwannomas. So when you see a schwannoma, chances are the NF2 gene is not working, right? That's kind of the hallmark of a schwannoma. It's mutated in 66% of the sporadic schwannomas. So remember, sporadic are the ones that you get after birth. So often when you do have these schwannomas, they're 66% of the time mutated NF2 gene. It's frequently lost in conjunction with loss of chromosome 22. So this is something very interesting. There's something about this disease that I don't really understand yet. Maybe they've talked about it, but I'm just not aware of it. But chromosome 22 is breaking, right, at the same time that you're often getting this mutation on gene 22. I don't think we know why yet. I'm not aware of that literature if it exists, but it is known that frequently these schwannomas have a damaged chromosome 22. It's not completely gone, but chunks of it are gone. Like, you know, one of its arms might be just totally absent, something like that. In Schwann cells, Merlin co-localizes with E-cadherin at the paranodes and Schmidt-Lantermann incisures in the myelinating peripheral nerve. That's a lot to digest. I'm going to move on because we have a picture of it. All right, so what we're talking about is you have nerves, and you have Schwann cells that are wrapping around the nerves. And I talked about when you wrap around the nerves, it's creating an insulator. It's kind of like the plastic around wires. You're insulating, but there is a junction. There's a gap between these insulating areas. That gap is, let me see, the Schmidt-Lantermann incisures. That's what it's doing from a biological perspective is you have what's called an action potential moving down the nerve. That action potential is the influx of those sodium ions. Now, what happens is sodium ions enter the inside tube of the axon, the long part of the nerve. When it's on the inside of the nerve, it changes the voltage, and that opens more sodium channels. Well, with more sodium channels open, more rushes in, and you get more voltage. And so you have a propagating effect where the sodium is actually pushing a wavefront of sodium ions, but also voltage potential along the tube. And that's the neural signaling. That's the firing of a nerve. Very quick, actually. Even quicker when you have that insulation. Now, that's great. You're insulating, but at some point the signal dies out, right? You can't insulate all the way to the other end of the cell because the signal is too weak, right? So every so often you have to have a break between the insulators where you have more of those sodium channels exposed. You're not wrapping it in fatty tissue. You're exposing it to the fluid, the extracellular fluid, and you're re-upping the ability for sodium to influx. And so those nodes are very important. They're an attachment place where the myelin is connecting and then stopping from spreading, right? And so that's what's pointed out here. So let me now, with that context, we'll go through this. So immunofluorescence analysis of these teased nerve fibers, which were from a mouse, are showing two proteins, AMOT and E-cadherin, which are junction proteins. They're for cell-to-cell binding. Arrowheads indicate the Schmidt-Lantermann incisures and paranodes. The paranodes are those junctions between the myelin. So you can see these lines here. That's where the in-between parts between those Schwann wrappings are occurring. So we're going to start introducing these terms. So one of the proteins were the angiomotin and the E-cadherin, which are cell adhesion molecules. Again, cell-to-cell binding. We have a picture of it coming up, but it's just cell-to-cell binding. And then what we're seeing is that that is also where the Merlin is co-located. So the Merlin, which is the red, and the E-cadherin, which is the binding protein, they're co-located. They're in the same place, and that place is these junctions on the myelin, well, in between the myelin wrappings. the paranodes. And so these, again, the Schmidt-Latterman incisures are cytoplasmic channels in the myelinated nerve fibers. Again, you're re-upping the sodium influx every so often. And you might also hear the node of Ranvier are the another name for those gaps, and we're actually looking at what's right next to it. So the paranodes are what's right next to those nodes of Ranvier. It's where it's on the myelin side of the myelin no myelin gap, right? You have this where the myelin ends, and after the myelin there's nothing. Before the myelin there's myelin, and then at that junction on the myelin side, that's what we're calling the paranode. Para means to the side of the node. All right, this is the main pathway as I'm going to explain it from a high level perspective. It's much more complicated, I think, and we will get into that. But I won't. This is as easy as I can make it, actually. And so this is the Merlin-Pack ERK pathway. It was one of the three that we talked about before. We talked about also the Hippo pathway. And let me go back. We talked about the mTOR pathway, the Hippo pathway. We're over here on this third one, RAC-Pack, because it proves the point well. It shows you why it is a tumor suppressor. So it's a schematic representation of the Merlin angiomotin RICH1 RAC1 pack signaling cascade, and how that relates to the RAS ERK pathway. So this RAS MEK ERK pathway, that's the same pathway from NF1. This is what ERK results in is cell growth, cell proliferation, and survival. So this is well known to be related to cell growth, right? That's the main pathway that a lot of disorders are compromising, including the NF1. You might recall that in NF1, the common treatment was a MEK inhibitor because they also had something over here to the right, which would have been promoting activated RAS, which was promoting MEK. So you had too much MEK in NF1, and so you were able to inhibit the MEK, inhibiting this kind of growth effect. Now we're up here at Merlin, and Merlin kind of has this kind of confusing. Just to be clear, the arrows generally are implying a promoting effect. So this promotes the next thing. The flat bar that looks like an upside down T is implying that's an inhibiting. So it gets a little bit confusing, but you can imagine if you're inhibiting an inhibitor, then you're promoting, right? And so there's a kind of a few steps of that, but ultimately what we're saying is that the function of Merlin, which well, I'll read it just here. The dotted curve reflects findings showing that Merlin can also directly inhibit PAC activity and in a negative feedback loop. PAC phosphorylates and inactivates Merlin. So this is common in regulatory signaling pathways where you have something turning something off, and that thing can also be turning its thing off, right? So it's a balance where they're both kind of inhibiting each other. So if we just look at the dotted one, if we know that increased ERK is increased cell growth, so the role of Merlin in this case would be to limit PAC, right? If we have healthy Merlin is inhibiting something that is promoting these other things, and those things are resulting in cell growth. So normally Merlin is suppressing the cell growth, right? It's playing that braking tumor suppressor role. It's a brake. If your brake is broken, if Merlin is not there, it's absent, it's defective for some way, then PAC is not getting that inhibitory signal. and therefore PAC is more likely to continue to add gas to the fire to keep promoting this MEK ERK pathway. So I want to take a minute to pause there and ask, is that clear? Because this understanding how, in this one case at least, Merlin is related to cell growth, it's very important. So I'm going to ask Arya. Arya, does this make sense to you?
Kind of, but still kind of confused too.
Sure, no worries. So let's see if we can simplify it. Okay, so I'm gonna see if I can. You guys can see. Well, actually, not on that one. Let me use a different tool. All right, so we have MEK, ERK, and then we have ERKs resulting in cell growth, proliferation, and survival. So what we're doing is we are just looking at the PAC interaction. So PAC is promoting CRAF and MEK. I'm just going to jump to say it's promoting MEK. So we have this PAK, was it? Yes, look, PAK. PAK is promoting this, right? Now, the healthy function of Merlin is to inhibit. So let me see what I can do here. We're going to make this one red and say inhibits, right? So if Merlin is triggered, you see if you can follow the logic. Merlin is triggered, there is less PAC. And if there's less PAC, then there is less MEK, right? Does that make sense so far?
Yeah, it does. Thank you.
Okay. And then, therefore, the downstream stuff, less ERK, less that. Now, conversely, if Merlin is broken, so we're going to put a big old X through Merlin. Merlin died. Sorry, King Arthur. We are killing Merlin. So your inhibitor is no longer effective. I'm going to change it from this solid line to this like kind of sad dotted line. It's not there anymore. Okay. A little bit more clear. So you have this sad dotted line. It's not there. That means that PAC is not downregulated. This is so I'm just going to put a not in front of it. So there's not less PAC, and there's not less MEK, and there's not less blah 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 blah. Right? So less ERK, and so you're losing a break in the system. Again, the proper term for that is Merlin plays a role as a tumor suppressor. So you need it to be there to act as a braking mechanism. And without it, you don't have the braking mechanism, and you have this propensity to kind of have tumors that grow out of control. Is that clear?
Yeah, that makes a lot more sense. Thank you.
Okay. Absolutely. Now this is very important. So I do want to ask a few other people. I guess Clara, Um, what happens to pack when Merlin is broken?
It overproduces and gets out of control.
That's right. You're not inhibiting anymore, so that yeah, you're right. It'd be like an upwards expression of it. There's too much probably. I'm just gonna continue, but before I do, who has any questions? This is very important. So who's not clear? Okay. So what I will say, just to make this, I do want to make this a little bit more complete and put it into context. So we talked about it coming in here at the RAF and the MEC. The reason you have this RAS and receptor tyrosine kinase is because that talks about what commonly is triggering that. So I'm going to go ahead and add that into our picture just for context. Where is the picture? Here's the picture. So I'm going to go back to healthy, normal-ish state. All right, so normally Merlin's there, it's doing its thing to turn things off. What also is normal is that you have the cell wall, right? So this is cell wall, cell membrane. And then you're going to have some kind of signaling happening. This is going to be, they're calling it RTK, that's receptor tyrosine kinase. You could also be, this is also the same conceptual location as EGFR, epithelial growth factor receptor. And of course, these things are docks, right? And so you're docking some little binding molecule. So in this case, you'd have epithelial growth factor, or you'd have, I don't actually know what binds to RTKs, but there's whatever, I'm just going to call it, it's probably a growth factor as well. There's probably some kind of growth factor binding there. Now, what do these do? So you say you have damage, right? You're going through that immune response. So there's going to be growth factors in the interstitial fluid binding to cells. This is normal. You want the cell growth and division because you have a wound, right? That's going to trigger the RAS. RAS is something that it's a very big orchestrating gene. It's kind of at the top of a lot of these pathways. There's a lot that's downstream of it. Let me just move this. So RAS normally is involved in essentially the promoting MEC, right? That's what's really what it ends up with. So I'm just going to follow the diagram. There is an intermediate CRAF, but ultimately it's promoting MEC. So you can kind of see where it fits in the normal healthy cell process. You have damage, you get growth factors. Growth factors are like epithelial growth factor, are binding to epithelial growth factor receptor, which are activating the RAS, which are activating CRAF, which are activating MEC, ERK, all the way down to cell growth, proliferation, survival. Generally, what that means is things that go into the cell and nucleus and activate things inside the cell nucleus. So the nucleus and promote protein expression. You're promoting the proteins that are actually going to start cell division in particular. So let me get my screens all set up here. So Suhan, what would happen in a normal cell to the cell growth if epithelial growth factor is injected near the cell? What would you expect to see? Suhan, you're muted.
Oh, sorry about that. I don't Let me think. Like, I feel like it would cause it to like grow and like divide like faster.
That's right. So you're having more of this input signal at the top, and that's triggering the downstream cascade. So yeah, it's a regulatory pathway, and now this also has to do with how you think about therapies. Now, I'm going to ask Anushka, if I were to have that situation where there's a lot of epithelial growth factor out here, and I were to introduce a MEK inhibitor, what would the role of the MEK inhibitor be? What would it do to the cell growth compared to without it?
If you had a lot of the epithelial growth factor,
And then you introduce a MEK inhibitor. What would the MEK inhibitor do?
I am kind of lost
Okay, no worries.
because, well, if you have more of the MEK inhibitor, does that mean that you get more like cell growth?
Actually, the opposite. So let's take a look at that. So the MEK is this one right here. So I'm gonna just highlight that in orange. So what we're going to do is we're going to introduce a compound that's a MEK inhibitor. Like for example, we know our good old what was it? Olivia from NF1. What was the MEK inhibitors? Selumetinib, was it?
Yeah, I'm pretty sure that sounds right.
Okay, selumetinib, a MEK inhibitor. So we're giving it a compound. So in this, I'm just going to get rid of these labels. This is being introduced at this stage. This stage is now being inhibited. So in the scenario, we had EGFR. So EGFR means that there is an overexpression here of RAS. There's an overexpression of RAC. There would have been an overexpression of MEK, but now we've added an inhibitor and we're stopping the cascade there. So we are able to give a drug at a later point in the cascade that could potentially be useful, right? And if we go back to maybe a more practical example, if we think about what if Merlin was broken again? We have broken Merlin. So what did we learn about broken Merlin? When we had broken Merlin, then PAC was inhibited, and that meant, or sorry, no, PAC was no longer inhibited, meaning that there was an overexpression of PAC. I'm going to say, instead of less PAC, we're going to say now we have more PAC. So we have more PAC. We would have more MEK, right? I'm ignoring, like, if you ignore the drug, right? Just not having Merlin means you would normally have more MEK. Except we do have the drug, except we have a downstream inhibitor that prevents the cell growth, signal propagation, prevents signal further signaling propagation. Olivia, does that make sense to you?
Yeah, I think so.
Okay. Anushka, does that make sense? How the MEK inhibitor kind of stops this cascade of things that were otherwise overexpressed?
Yeah.
Okay, cool. So
That
that's
does.
basically the role of therapies, right? When we're looking at Merlin is broken, we're probably going to be looking at things that are downstream of Merlin, somewhere in the downstream cascade that we can stop or do something with to prevent that signal. Now, where downstream, that's the million-dollar question, right? Which one of these do we want? Well, it has to do with what's effective, what's the side effects profile, but it fundamentally needs to be somewhere in one of the known or one of the biologically relevant pathways. And either we know that pathway or we don't know that pathway, but it's a real pathway that the cell must have and be using actively. Go ahead, Anishka.
Can you go back to the diagram that you were showing us?
Yes. Second, here it is.
I think you asked me like if you introduce like the EGF, the growth factor. I just wasn't sure like where does that like come into play?
It was a trick question in a sense. So what I was saying was by having a lot of EGFR, right, then we would expect a lot of CRAF, right? We'd expect activated CRAF, I'm implying here, activated. So we would expect that. Now, without the inhibitor, we would have expected a lot of activated MEK, right? But because we have an inhibitor, we're stopping that cascade at this point in the pathway. So that's why it was a little bit of a trick. Without the MEK inhibitor, we would have thought that yes, there would be absolutely an increase in the ERK and all the stuff downstream of it. But by introducing a specific type of inhibitor at something downstream in the sequence, we're stopping that signaling from going any further.
Okay, yeah, that makes sense.
Okay, cool. All right, let's go back then to the deck. So again, I don't expect you to memorize this. I want you to have kind of a passing familiarity with some of these acronyms. But you'll go into them more over the course of this year, I think. And by the way, this is one of the three that we talked about. So that's just the RAC pathway. There's also Hippo and mTOR, which I don't have fully documented, but we can look into those. All right, so going back to Merlin, it's also involved in other pathways like the mTOR regulation. It's also shown to be in the Hippo pathway. And one of the things it's doing is it's playing a role in membrane recruitment and activation of the RAC pack. We already know about the activation of the pack. If you know, Merlin's not there, then pack isn't being inhibited. So it has to do something with that. What we haven't talked about too much is membrane recruitment. And that's actually what a lot of these slides coming up are. We did talk a little bit about co-location, this idea that not only do proteins need to exist, they need to exist at the right place. And if they're playing a role with other proteins, then they need to relate in the same spot. So if there's something about what myelin is doing at those nodes where it is like connecting to the nerve, but also not going further into that node of Ranvier, that junction area. There's something about how it's gluing the myelin together with the proteins of the nerve. And we have a picture of that coming up, but just keep that in mind that there's something about membrane recruitment that Merlin is doing. It's playing an orchestration role structurally with how proteins and also cell-to-cell connections are being made. So we do know that upon cell-to-cell contact, Merlin plays a role in downregulation of epithelial growth factor receptor. So you're saying literally there could be less receptors because of something Merlin is doing. So again, it's playing a role not only in cell-to-cell connection, but also what proteins are being recruited from the cytoplasm into the membrane? What are we squishing into the membrane and you know, locking into place? We're playing some kind of role in that as well. So Merlin has been found to suppress tumorigenesis by entering the nucleus and binding to this E3 ubiquitin ligase, suppressing its activity. We kind of touched on that before. It has this other thing it's doing. And for translocation into the nucleus, Merlin must be activated. So it's a specific type of Merlin. It's activated configuration, and it must be activated by MYPT1, myosin phosphatase target subunit 1, although other mechanisms of action could not be ruled out. So I wouldn't say it's the primary driver of the tumorigenesis. However, it's one of the pathways, right? There's one of the problems here with Merlin is that it touches multiple systems from multiple places. It touches cell growth through the PAC pathway, as we saw. It also touches it through the Hippo pathway. It also touches it through the mTOR pathway. It also touches it through this ability to enter the nucleus and do something there. And so that's something that, you know, when we talk about where would a therapy be? Well, a therapy might be downstream of any one of those 4 pathways, right? And maybe, this is just purely hypothetical, but maybe you have a version of the disease where one of those pathways is particularly relevant for you. Maybe the PAC pathway is the dominant pathway for you, and for somebody else, the Hippo pathway is very dominant. And to make a connection for you, if we go all the way back here, you kind of get that sense also where, you know, what is the driver of the schwannomatosis? It depends, right? There's a little bit of variability there. We are focusing only on the top one, though, this NF2 schwannomatosis. And what I'm saying is in that one, there are still 4 things that could happen. Those 3 main signaling pathways, and then this nuclear migration translocation effect. And so it can be challenging to find which one works for the most people on average. Or in the best case scenario, in truly personalized medicine, we would know which of the 4, or if it's all of the 4, that are driving the tumor growth. You know, there's an actual answer in there somewhere. If you were to really, really study a single person's cancer for a very long time, you could actually get an actual answer on what's driving their specific tumor growth. All right, so now we're going to kind of jump off the deep end and go a little bit even more complicated, but I promise you I break this into parts. So what we're going to do, just to kind of give you an idea, these are pictures of what the Merlin is doing. Merlin is one of the proteins in this structural protein diagram. So the black line is the cell wall, and we're going to talk about the tight junction and the adherent junction. These are 2 ways that cell-to-cell binding is happening. And what you see is that where these cell-to-cell binding proteins are, that there is a lot of co-located proteins doing stuff, including Merlin is co-located with these junction cell-to-cell wall binding proteins. And what we're going to go through is there is a state where there is healthy, so like this growth suppression condition, what's saying is that's when Merlin is doing what Merlin's supposed to do, which is to play that inhibitory role. So it's growth suppression and then it's configured like this. But in that tumor state where you have Merlin is playing a growth stimulation function, then it would look like this bottom graphic. So I'm going to break these graphics into a few slides. We'll go into what all the acronyms mean, and then we'll circle back and we'll actually talk about what's actually going on in these pictures. But for now, just be aware that you don't have to understand. I know it's very overwhelming, these graphics. we're going to go into it. I'm just going to read this. Merlin inhibits RAC1 activity by releasing RICH1 from its tight junction associated inhibitor, angiomotin. So what's shown here is a model depicting the proposed mechanism of how different interactions between Merlin, angiomotin, RICH1 are modulating that RAC1 activity. You may recall RAC1 is this right here. So RAC is upstream of all of it's upstream of MEK and ERK, these things that result in tumor growth. So, you know, we want to turn RAC off because RAC is promoting cell growth. And normally Merlin is playing this dotted line, this big black one here, of inactivating the RAC. But in the broken case, you see that the dotted means it's not doing that. We're not inactivating the RAC, right? So that's why we're getting the cell growth. So under growth suppressive conditions, that's the healthy one, Merlin binds to angiomotin, releases RICH1 to inactivate RAC by converting RAC GTP, that's guanine triphosphate, that's literally 3 phosphate groups connected to a guanine amino acid base pair, which is actually on a protein. This is a very common way that you're turning proteins on and off, is by adding a phosphate group. So we can see the definition that RAC in its triphosphate state is active. In its diphosphate state, two phosphates, it's inactive. And so the functional thing that we're actually doing is we're controlling when this phosphate group gets added. Adding the phosphate group changes the shape of the protein. Usually it's an enzyme, and that makes it like turn the enzyme on, basically, so it's able to do its signaling and kind of do a lot of chemical reactions. So in response to growth stimuli, Merlin disassociates from angiomotin. I'll leave it at that. We'll get into it. All right. So what we're going to talk about here are epithelial cell-to-cell junctions. Now, keep in mind that we are talking actually about Schwann cells. Schwann cells are not exactly this. The model was made for cells like the ones that line your blood vessels, like your capillaries. So there's generally an inside, that's this basal surface where the tissue is. The apical surface is where the fluids are. And so you have these like little cilia things interacting with the fluids. So even though we're not talking about the same cell in the picture, they are very closely related from an evolution perspective or a developmental perspective. As your cells are dividing and they're differentiating, the epithelial cells and the Schwann cells are very closely related from a developmental perspective. So this is still very relevant conceptually for what we're doing. So, and again, these are terms from this graphic. We talk about apical surface here. That's why I bring it up. So you have the apical surface, and then you have tight junctions. So tight junctions are, as you can guess, very tight. They form an apical seal between the epithelial or endothelial cells and maintain apical basal polarity. So they are tight. They are creating a seal, like a watertight seal. You don't want your blood leaking everywhere, and they're also maintaining a polarity. So look at this. There's an apical side and a basal side. The tight junction is going to be only towards the apical side. We are maintaining a polarity by recruiting things to one surface of the cell that we're not recruiting to the other side. So things that are forming these cilia, for example, they probably need to be recruited to this side, not the inside, right? So they play a role in the polarity inside the cell. So the cell is obviously implied to have an orientation. The tight junctions restrict paracellular flux. So a cross-cellular flux is like a movement of fluid, and serve as signaling hubs that link membrane receptors to the cytoskeleton and transcriptional transcriptional programs. We talked about where do the membrane receptors go? Well, imagine that you're sampling things from the blood. Which side do you want your blood receptors to be on? You want them on the apical side. Well, how do you do that? Well, one of the ways is you could be involved in these tight junctions so that if you co-locate near the tight junctions, you're co-locating near the apical side. You have the receptors on the blood side, not on the basal side. So it's doing stuff like that. That's probably an oversimplification. The body's very complicated, but you get the idea, right? Like we are using the structural scaffolding of the cells, and the scaffolding is maintained and preserved by where certain proteins are, and especially these structural ones. And so that's the tight junction. There's another type here. You also, by the way, look at these tight junctions. You see how they weave back and forth, and they're actually connecting between the two cells, right? So you actually have a strong interaction between these proteins. Adherents are also strong, but you see how they're stacking on top of each other. It's still strong. It's just maybe not as strong as this weaving pattern. But the adherent junctions lie immediately basal to the tight junctions. That's below in this picture. They're basal. And they mediate strong cell-to-cell adhesion and connect cadherents to the actin cytoskeleton. Please recall that inside your cells is actin, and it is like a highway of fibers, and these are rigid fibers, and they connect within your cell so that things can like move around your cell, but also across your cells, right? So like you have that extracellular matrix. Everything's connected. There's this connective tissue, and that it's actually connected at the protein level with these E-cadherins right here. So that's the adherent junctions. They regulate tissue integrity, mechanotransduction. So anything you have any touch receptors, anything that the vibration matters, they might be playing a role in that. Contact dependent control and of proliferation and migration. So, if you need to touch something for it to start healing, something like that. All right, so let's just zoom in on the tight junctions. And this is calling out from our graphic where the tight junction is. It's on the right. And you see this PDZ is the same thing as this this weaving pattern up here. So the tight junction modules and domain architecture. So there's a PDZ scaffold protein. There could one or many, right? There's multiple PDZ domains. They're in this ZO family. Okay, just to connect those. And they assemble transmembrane claudin occludins and recruit signaling effectors. And that's these things that they're connected to up here. The PDZ array provides multivalent docking. And that's just a way of saying that many things connect to these PDZs. You see all of these different things up here that are connecting. A lot of things are interested in being connected near these tight junctions, and they have a dock that lets you dock to it, and a lot of things are using that style of dock. The L2Z domain, and we're seeing that up here, mediates homo and hetero oligomeric assembly of polarity complexes, helping build higher order junctional scaffolds. Something to do with how it's, again, polarity. It's located in a specific side of the cell. Something to do about higher order structural stuff. Again, polarity was the word we were using before. We're seeing that there's this Crumbs apical complex. Crumbs family proteins Crumbs family proteins localize the complex to the apical surface and help define the apical membrane domain. So Cribs has this particular function of pointing this whole big old structure. And then PALS 1 module, where is that? This thing over on the left. It's some of those things that are docking on this, and they link crumbs to cytoskeletal and membrane trafficking machinery, and it acts as a key organizer for apical polarity. So again, Merlin is interacting somewhere with things that have to do with how the cell is moving things to relevant parts of the surface of the cells, and specifically the apical surface, right? The one that's pointing outside. So we just, that's all we need to know for now, but we kind of keep that in mind. Like there's something structural going on here, and it's playing a role in it. So now going down a little bit tighter, we have this adherent junction. The adherent junction, you'll see it's co-located below the tight junctions, has this kind of interaction here. So the angiomotin, shown as AMOT sometimes, but we see that here. This angiomotin is a coiled protein that localizes to junctions, scaffolds polarity and hippo pathway components, and binds the F-actin and small GTPase regulators. So we see that's binding here to this rich one. This gap is it's a GTPase, which ACE is enzyme, and a GTPase is something that is adding or removing GTPs. And we see that here. It is the thing that is adding the 2-rack a GTP to GDP. I think this is the dotted line. This is the broken one. But what it would normally be doing, this rich one, and the gap domain in it would be adding that phosphate group. And so we see that it's tied to this angiomotin, tied to the junction near Merlin. We haven't quite gotten to Merlin yet. All right, so Merlin is a firm domain protein that links membrane complexes to the cortical cytoskeleton. It localizes to both the adherent junction and tight junctions and associates with angiomotin and polarity scaffolds. Firm domain interactions. Firm domain in Merlin mediates binding to transmembrane receptors, cytoskeletal linkers, and junctional complexes, enabling contact inhibition signals. So again, it plays a role with the receptors, plays a role with the things that touch the cytoskeleton and these junctions. It's very structural, and it's where it's located and what it's doing. We see the firm is part of the Merlin, and it's touching this junction here, co-locating. Its function in part is to play a role as a co-locator. If you don't have it there, things aren't connected. The Legos aren't in the right place, right? So one of its roles is this co-location effect for receptors, but also the signaling machinery here, angiomotin they're talking about, which has this rich one in the gap and all that other stuff. By the way, this is all, when you have multiple proteins that come together, it's called a protein complex. They're functioning as one unit, even though they're made of separate proteins. You see there is a cytoplasmic tail, CT, of cadherins that recruits catenins and tight junction adapters that communicate with firm and actin. Though there's a tail here, part of the regulatory process, kind of detailed. We don't need to get into it too much. Bar domain and rich one. Bar proteins sense and induce membrane curvature and help traffic junctional proteins. Rich one is a gap protein that links junctions to RAC1 regulation. Again, the function of this, despite how it is and where it is, the function is to interact with RAC to turn it off. So Merlin is positioning these other things to turn it off. Without it, they're not positioned. It's never turned off, and you have this out of control system. Okay, so yeah, going back to this, hopefully this now makes a little bit more sense in context. So again, on the left, we see that Merlin is needed for these other things. It's needed for the angiomotin, for the Rich1, and without Merlin, you're not co-locating these things in the right place. And without that, then you're never able to turn RAC off. And without that, then you just follow this cascade on those bottom left. Without RAC turning off, then PAC is out of control, MEC is out of control, and ERK is out of control. You have tumor and cell growth. So that's a lot. And the reason I went through it so deeply is because this disease is so poorly understood from a drugs perspective that we really need to understand. If we're proposing a drug, we want to know where in the pathway, in the cascade, does this drug live? This, I think, covers very well the PAC pathway. I have not shown the Hippo pathway. I have not shown the mTOR pathway. And I have not shown the nuclear activation pathway. But that's the idea, is that especially if we're trying to propose something that is not really well understood, we do have to understand the pathways. And you can't really do the drug development in earnest until you have some kind of pathway understanding inside the cell. But thankfully, that documentation does exist in literature. It might be a little bit disparate. We might need to comb through it, but there's a lot of people studying what's going on inside the cell. But putting it together with drugs and drug outcome data, which we have, that's going to be very novel and very important for us. So I'm sure you're confused. That's totally okay. Again, my goal is to try and introduce it to you now so that the more you encounter it, the more familiar it becomes. We probably will go through this one or two more times. What we have coming up at our next session is we have a data set that has drug outcomes data. So I'll bring Paul on board. He's one of our senior scientists. He will go through the data, some of the possible algorithms we can start to look at for sorting that data. Once we have data, we run it, we rank it, we get our top compounds. We want to then take those top compounds, go back to this picture and say, why? Why was this drug that's a MEK inhibitor useful? Well, okay, I know it's useful because it's downstream. I can see it right here in the picture. It's downstream of what's going broken, and we're doing the opposite thing. So that's generally what we need to do, is we need to have a rationale for why compounds that we're finding to be effective are probably what they're doing, what their effectiveness is. So that requires us to learn more about the key pathways involved. We should probably look closer at the Hippo pathway and the mTOR pathway. I'm happy to kind of put some slides together for that coming up. But also on your guys' side, doing the literature review, that's going to be very important.